IB Bio Genetics Part 8 has its focus on a statistical approach toward understanding the outcomes of crosses in genetics. In the same way that the t-test allows us to determine significance between two means, the chi-square allows us to analyze relationships between two groups of data, observed and expected values. Chi-square analysis can be used in genetics or behavior experiments. The material here is for HL students only, but SL students may need to use the chi-square analysis for IA work. The essential idea is genes may be linked or unlinked and are inherited accordingly. Here is the outline of all the genetics movies available for your study. Use this outline to find the movie you need. This movie is focused here. Let's start with an example. Suppose you plan to flip a coin 100 times. You would expect that 50% of the tosses would produce heads and 50% of the tosses would produce tails. Upon completing 100 coin tosses, the observed results are 47 heads and 53 tails. Now, what's going on here? Why not 50-50? Is the coin weighted? Is the coin not a true coin? Now, you're forced to examine the difference between observed and expected carefully. Is the difference large or small? Each flip of the coin is an independent event, and whether or not it lands on heads or tails is random chance. With 100 independent tosses, we would expect numbers to be close to 50-50, but not exactly 50-50. Now, are the differences between observed and expected large enough to suggest a weighted coin, or are the differences small enough to assume that random chance caused the differences? The chi-square analysis allows us to be more certain about our conclusion. While the differences between the observed and expected values on the last slide were most certainly due to chance, how far away from expected values would our observed values have to be before we suspected something other than chance was going on? Here you can see numbers that stray far from expected values. Is the difference between observed and expected due to chance? It's not so easy to know. Is the coin weighted? Is something other than chance at work here in the relationship between heads and tails? How would we know? Well, the chi-square analysis helps us assess whether the difference between observed and expected is due to chance or due to some other factor requiring another explanation. Here is the first IB syllabus statement to kick off the movie. Use the chi-square test to determine whether the difference between an observed and expected frequency distribution is statistically significant. Here's the chi-square formula. This is the chi-square value. It will be a number. This is sum of, I'll come back to that, observed minus expected squared divided by expected. That comes from the data, and we would do this calculation for both heads and tails and sum them together to get the chi-square value. So I've come back to the coin flip data, the data with results that were striking because the observed values were far from the expected values. We will calculate a chi-square value here that will be plugged into a probability table, I'll show you that on the next slide, to provide a probability value, the probability that the difference between observed and expected is due to chance. Now, here's the chi-square formula, and I've calculated components in the table. Here is the observed minus expected for heads and for tails, observed minus expected squared for heads and tails, observed minus expected squared divided by expected, that value's here for heads and tails, and then we sum the two values together to get the chi-square value of 4.84. Now, what does this mean? I must put this number up against a chi-square probability table to determine its meaning. So we plug the chi-square value 4.84 into a probability table seen here. Now, before we do that, there are a couple of things to explain. With our chi-square value, we must go into the table along a row. It's called a degrees of freedom row, and the question is which row? Degrees of freedom is related to sample size, or in the case of chi-square, how many classes were involved. In our analysis, there were two classes, heads and tails, and we subtract 1 from 2, so we're going to enter the table on row 1. The subtraction of 1 from the number of classes is a detail of statistical tests that we don't need to go into here. Just remember to count your classes carefully and subtract 1 and then enter the table on that row. In this example, we come in on the first row because we only had 2 classes, heads and tails. 4.84 fits in right here. We read up to the probability value, which falls below 5%, the probability that the difference between observed and expected heads and tails values is due to chance less than 5% of the time. So the probability that the difference between observed and expected is due to chance is low. It's less than 5%. Therefore, some other explanation is needed to explain the results. They're not due to chance. Is the coin weighted? Now, just for clarity, let's go back to the results that produced 47 heads and 53 tails for chi-square analysis. If you run the chi-square calculation, which I'll let you do on your own, the chi-square value is 0.36. We enter the table on the first row. Again, we subtract 1 from our 2 classes, heads and tails. We come in on the first row. We come over to 0.36 and read up to the probability value. The probability value is between 50 and 75% due to chance. Thus, the probability that the difference between observed and expected being due to chance is very, very high. Thus, the difference is not significant. We can conclude that the difference between observed and expected is due to chance, nothing more. Like coin tosses, genetics experiments have expected outcomes. Like coin tosses, observed values are not likely to match up with expected values exactly. But the same question stands. Is the difference between observed and expected due to chance? Chance in genetics comes from random fertilization, random mating. We do not know which sperm has reached which egg. Let's try our hand at chi-square analysis in genetics using a cross of fruit flies, gray body fly, against an ebony body fly. The experimental hypothesis is that the pattern of heredity for the gene for body color is a Mendelian dominant recessive pattern. The F1 will show a 1-0 phenotypic ratio, gray to ebony, and the F2 would be expected to show a phenotypic ratio of 3 gray to 1 ebony. There could be a different experimental hypothesis, one that examined sex linkage, where we would count the numbers of males and females with the trait. But let's keep our focus on the first experimental hypothesis. Here are the numbers for the gray body fly crossed with ebony body fly from parents all the way through F2. Notice immediately that for the F1, 100% gray was expected and 100% gray was the result. If the result came out any other way, we would start over with a new hypothesis. Literally, we would back up and begin with a different experimental hypothesis. But in this case, we can move forward. Notice the F2 shows large numbers of gray and smaller numbers of ebony. Are the data supportive of the 3-to-1 expected ratio, or is some other heredity pattern at work? We don't really know until we run the chi-square. Lastly, notice that male-female numbers are close to each other, suggesting that the gene is not X-linked or Y-linked. And remember that the null hypothesis comes from the probability table that the difference between the observed and expected results is due to chance. So let's focus our chi-square analysis on the dominant recessive hypothesis rather than the male-female hypothesis. If we were interested in determining sex linkage, then we would want to conduct the chi-square analysis on differences between males and females. We're looking at the F2 gray versus ebony numbers to determine if the gene operates in a Mendelian dominant recessive pattern. And here's the chi-square formula that we're going to use. Here is the same data that I presented 2 slides back, but in this slide I'm only presenting F2 data, and I've combined males and females to get a total number of gray flies and a total number of ebony, and the total number of flies counted being 456. And I use 456 to determine my expected values of 342 and 114. 342 is 3/4 of 456, and 114 is 1/4 of 456. And I take the calculations in parts: observed minus expected for gray, observed minus expected for ebony, observed minus expected squared gray, observed minus expected squared for the ebony, observed minus expected squared divided by expected for gray and for ebony, and then summed to get the chi-square value of 1.68. Then I take this value to the chi-square probability table. Remember, we have two classes here, just gray and ebony, so we'll be entering on the first row. So we enter the table with 1 degree of freedom. Remember, two classes of phenotypes, gray and ebony, minus 1. We find where 1.68 fits and then read up to the probability value. The probability due to chance is high, higher than 5%. Therefore, there is no significant difference between observed and expected. We accept the experimental hypothesis that gray is dominant to ebony and that this gene operates in a simple dominant recessive pattern. Now, if the difference between observed and expected was large, resulting in a low probability value due to chance, then we would need to start the statistical work over with a new hypothesis formulated around a new pattern of heredity. Now let's run a chi-square analysis on an X-linked cross in fruit flies where you have a red-eye female crossed with a red-eye male. But imagine that you don't know anything about sex linkage, but you suspect as much, so you develop a hypothesis. The gene for eye color is located on the X chromosome. The female is a heterozygote. This is part of the hypothesis. And you anticipate offspring with the ratio seen here. This is all part of your hypothesis. So the experimental hypothesis is that the pattern of heredity is a sex-linked pattern. The cross is a red-eye heterozygous female, again this is a hypothetical, and a red-eye male, so that the F1 would be expected to show the following phenotypic ratio: 2 red-eye females, 1 red-eye male to 1 white-eye male. Now, if you really didn't know anything about the heredity pattern of the gene, you step in with a hypothesis like this, ready to erase and start again if the chi-square analysis does not support the hypothesis. It's that simple. The null hypothesis, of course, comes from the probability table that the difference between the observed and expected results is due to chance. Here are the numbers, the observed phenotypes, with a total count of 212 flies. We use 212 to determine the expected values. We expect approximately half of 212 to be red-eye females, approximately a quarter of 212 to be red-eye male, and approximately a quarter of the 212 to be white-eye males. We run the numbers using the chi-square formula to produce a chi-square value of 1.92. I'll let you do work on your own to confirm the calculation of 1.92. Stop the movie now and work through the process so you're confident in the calculation of the chi-square value. We enter the table with 2 degrees of freedom because we had 3 classes of phenotypes. So 3 minus 1 has us entering on the second row, moving across to 1.92, coming up to the probability value, which is large. It's above 25%. Thus, the probability due to chance is high. There is no significant difference between observed and expected values. We accept that the gene is X-linked and that the female was a heterozygote. So here's an IB syllabus statement that takes us further along in our chi-square analysis. Use the chi-squared test on data from di-hybrid crosses. Here's a di-hybrid cross. We're going to identify this gene as seed shape, round and wrinkled, and this gene seed color, yellow and green. Here's the experimental hypothesis. If the 2 genes are not linked and behave in a dominant recessive manner, then the expected phenotypic ratio from the di-hybrid cross would be 9 round yellow, 3 round green, 3 wrinkled yellow, 1 wrinkled green. Let's see what the numbers are. Here are the observed values, totaling 556 seeds counted, And the expected values come from the 556. 312.8 is 9/16 of 556. 104.2 is 3/16 of 556, and this is 1/16 of 556. The chi-square calculations are run, producing a chi-square value of 0.477. Turn the movie off now to confirm this value. We enter the chi-square table with 3 degrees of freedom because there were 4 classes of phenotypes. The probability due to chance is higher than 5%. There is no significant difference between observed and expected values. We accept that the genes assort independently and behave in a dominant recessive manner. Let's look at other types of experiments where the chi-square analysis can be used and pay close attention to the hypotheses and the analysis. Behavior experiments can be conducted where organisms are observed in controlled conditions and position data as counts are collected over time. For example, an experimental hypothesis might be that the insects will be found more in location A because, again, due to some independent abiotic variable. The null hypothesis would be that the insects will be found equally among all locations, and this relates to the null hypothesis for chi-square that the difference between observed results and expected results is due to chance. Here are the number of counts in different locations within the habitat. And using the total number of counts, and I've made this very simple, we determine the expected values. We run the chi-square calculations to determine a chi-square, which is the sum of these 4, chi-square value of 4.4. With the chi-square value of 4.4, we enter the probability table on the 3rd row. We had 4 classes, and we determine that there is a high probability due to chance. There is no significant difference between observed and expected. The differences between the classes is due to chance. We reject the hypothesis that the insects will be located more in location A than in other locations. The abiotic factor used to explain the experimental hypothesis is not important to the position of the insect. We accept the null hypothesis that the insects will be found equally among all locations. So here's a chance to practice. Let's assume we're going to cross 2 tall plants against one another, and that all the offspring are coming from these 2 tall plants. And we're going to propose an expected phenotypic ratio. You can see down here it's going to be 3 to 1. So here's my experimental hypothesis, that the pattern of heredity for the gene for plant height is dominant recessive. Both individuals are heterozygous, that's a hypothesis, and thus the expected phenotypic ratio will be 3 to 1. And any differences in between classes would be due to chance. Now you might say, did you just guess about the expected outcome here of 3 to 1? And my answer is yes. I just proposed a hypothesis and I'm ready to start over. I'm going to work with a pencil and eraser, and if the chi-square analysis does not support this hypothesis, I start again with a new hypothesis. Here are the observed numbers. Here's the total number of plants counted, and I've left the table blank so that you can practice. Can you calculate the chi-square value? Stop the movie now to do so. Here's the completed table and the chi-square value of 0.607. We enter the table on row 1 to 0.607. We come up to determine that the probability due to chance is higher than 5%. There is no significant difference between observed and expected values. We accept the experimental hypothesis that the parents are heterozygous and that the alleles behave in a dominant recessive manner. And that brings us to the end of IB Biogenetics, Part 8.